Good afternoon, everyone. My name is Michelle File, and today I'm going to talk to you about an enzyme called amylase. So today, let's say you ate a large meal full of different kinds of starches. Did you ever wonder how your body breaks those starches down? Well, today I'm going to talk about the enzyme amylase that helps us do that. So glucose is a main energy source for our body. However, glucose from our diet is more likely to be found in much larger forms of sugars, for example, fructose, lactose, and sucrose, and it is essential for our body to be able to break down those large molecules into individual glucose units for our body to digest, and this is where amylase comes into play. Amylase, specifically alpha amylase, begins the process of digesting starch. It is an enzyme that is made in two places. There's the salivary glands that make salivary amylase, and that is the enzyme that ultimately breaks down starch, and this begins the digestive process. The second place is the pancreas, and that makes pancreatic amylase. This passes through a duct that will then enter the small intestine. The purpose of the pancreatic amylase is to complete the digestion process of carbs, producing glucose. Both the salivary amylase and pancreatic amylase are known as isoenzymes because they are enzymes that differ in amino acid sequence but catalyze the same chemical reaction. A significant function of salivary amylase is that in the absence of pancreatic amylase, salivary amylase could serve as an alternate pathway for the digestion of amylose, amylopectin, and glycogen. This is especially important in diseases like chronic pancreatitis and cystic fibrosis, where the levels of pancreatic amylase are very low or absent. Both salivary and pancreatic alpha amylase are considered chloride-dependent molecules that require the binding of a chloride ion to be allosterically activated. Amylase requires the chloride ion to be bound to the binding site in order to become active. The binding site has a positively charged amino acid residue that couples with the negatively charged chloride ion. This binding causes a conformational change to the enzyme that will allow it to actively bind to starch to begin the breakdown. The two figures shown are examples of how a specific large molecule gets broken down. In the figure on the left, it is comparing the pocket of amylase to the pocket of cellulase. Amylase specifically binds to starch, whereas cellulase binds to cellulose. Amylase is unable to bind to cellulose because the pocket is very specific to starch, and vice versa. The figure on the right shows the same thing. The top digesting enzyme is cellulose, binding to cellulase, and the bottom digesting enzyme is amylase, binding to starch to break it down to a more simple molecule like glucose, so our body can utilize it. Alpha amylase catalyzes the hydrolysis of an alpha-1,4 glycosidic bond in starch, one of the most abundant molecules on Earth essential to plant and animal energy storage. The structure of both human salivary amylase and human pancreatic amylase are found to be 496 amino acid residues long. The salivary and pancreatic alpha amylases are highly homologous in terms of primary sequence, but do exhibit somewhat different cleavage patterns. The largest amino acid change occurs in the segment of polypeptide chain that connects domains A and C. When comparing their amino acid sequences, there are 15 amino acid differences between the two sequences. For the structure of human salivary amylase, it folds into a multi-domain structure consisting of three domains, A, B, and C. Domain A has a beta-alpha barrel structure, domain B has no definite topology, and domain C has a Greek key barrel structure. There are 5 disulfide bridges that help form the tertiary structure as well. Similarly, human pancreatic alpha amylase is found to be composed of 3 structural domains. As in salivary amylase, the largest domain is domain A, which consists of an 8-stranded parallel beta barrel structure surrounded by a cylinder of alpha helical segments. Domain B is the smallest domain and primarily forms a beta structure. It is domain C that is found to be the most variable between alpha amylases of different origins. It is made up of antiparallel beta structure and is only loosely associated with domains A and B. The theoretical or computed molecular weight for alpha amylase was estimated to be about 57.7 daltons. Compared to the actual molecular weight of 56 daltons, the theoretical was fairly close. The isoelectric point is the pH at which a particular molecule carries no net electrical charge, and the theoretical or calculated value I got of the isoelectric point of amylase is 6.60, which is also fairly close to the actual isoelectric point, which is 6.34, meaning that at neutral pH of 7, amylase has a slightly negative charge. Cello predicts that this enzyme is located mainly in the extracellular space of the cell, which makes sense because amylase needs to be able to bind to the chloride ion in order to be activated. To get an idea of how closely related the nucleotide or protein sequence of amylase is in humans compared to various organisms, I conducted a percent identity matrix. I took the sequences of a bird, fish, chicken, mouse, cattle, rabbit, goat, and dog, and used clustable omega to calculate the percent identity matrix. Column 1, you can see that 100% is the first percentage. That tells us that we are taking the bird protein sequence and comparing the rest of the organisms to that. So wherever there is 100% in a column, that is how we know which organism's protein sequence the rest of the organisms are getting compared to. And I listed the organisms below each column. For the human amylase enzyme, we can see that the dog is most closely related to it with a percent identity of 90.41. We can also see that the organism least closely related to the human protein sequence of amylase is bird, with only 21.72% identity. To lay out this information of relativity a little differently, I made a phylogram. This confirms that the human and the dog are most closely related on an evolutionary scale. You can see that the chicken doesn't seem to be related much to any other organism on the evolutionary scale because the line doesn't have a distinct relationship with any other organism. Interpro, domain, and family analysis tells us that alpha amylase is a part of the glycoside hydrolase family. Amylase acts on substrates containing an alpha-glucoside linkage, so this makes sense that it belongs to this family. Amylase also has an alpha-like structure with a catalytic core. So next time you are hanging out with your friends and eating starch-containing food, just remember that the enzyme amylase is working hard to break down those complex molecules for you.